The hours between your last meal and your first morning breath are not empty hours. They are among the most biologically active, most physiologically consequential, most quietly extraordinary hours of your entire day, and what you choose to eat before you enter them, particularly after 60, is not a trivial decision about late-night hunger. It is a decision about what your brain cleans, what your muscles repair, what your heart recovers, and what your body becomes by the time morning arrives. There is a particular kind of ignorance that is not born of carelessness, but of assumption, the comfortable, culturally reinforced assumption that because something has always been understood in a certain way, it is being understood correctly. The assumption that sleep is rest, that the hours of darkness represent a biological pause, a temporary shutdown of the body's activity, during which nothing of metabolic consequence occurs and nothing of nutritional significance is required, belongs precisely to this category of innocent but consequential misunderstanding. And for people over 60, whose bodies are performing some of their most critical and most irreplaceable biological maintenance work during the very hours that this assumption has led them to treat as nutritionally irrelevant, the cost of that misunderstanding is paid not in any single dramatic episode of harm, but in the quiet, cumulative, compounding biological deficit of nights spent attempting extraordinary physiological work without the raw materials that work specifically and urgently requires. The body that closes its eyes and enters sleep does not slow its biology. It redirects it, shifting from the outward-facing metabolic priorities of the waking day toward an inward-facing program of repair, renewal, consolidation, and maintenance, whose sophistication and biological importance exceed almost anything the waking body accomplishes across the same duration of time. This redirected biology is not random or opportunistic. It is precisely orchestrated, stage-specific, hormonally coordinated, and exquisitely sensitive to the nutritional environment in which it must operate, which means that what a person eats in the hours before sleep does not merely influence how comfortable they feel as they drift off, but shapes the biological conditions in which every process of nocturnal restoration either proceeds with full effectiveness or is compromised by the absence of the molecular substrates it cannot function without. Growth hormone stands at the center of the sleeping body's anabolic activity, with an importance that its relatively obscure status in the popular health conversation dramatically understates. The largest single pulse of growth hormone secretion across any 24-hour period occurs during the early stages of deep slow-wave sleep, a surge that dwarfs the modest daytime secretions that occur in response to exercise and nutritional stimuli, and that directs the body's repair machinery toward a breadth of tissues and biological processes that no waking intervention can replicate with equivalent completeness. Skeletal muscle protein synthesis, stimulated by growth hormones' activation of insulin-like growth, factor 1 in peripheral tissues, peaks during this nocturnal window, repairing the microtrauma of the day's physical activity, rebuilding the protein structures that metabolic turnover has degraded, and contributing to the maintenance of the muscle mass, whose progressive loss after 60 is one of the most consequential and most practically devastating biological changes of advancing age. For this growth hormone-driven synthesis to proceed with anything approaching its full restorative potential, the amino acids it requires must be available in the circulating blood, and in a body that has not been nutritionally supported in the hours before sleep, that availability is precisely what the overnight fasting state systematically and progressively, the glymphatic system, the brain's dedicated waste clearance network, whose existence was unknown to science until relatively recently, and whose importance for long-term neurological health is now understood to be among the most significant discoveries in modern neuroscience, performs its most complete and most consequential work during the same deep sleep stages that growth hormones' nocturnal secretion requires. During waking hours, the brain's glial cells maintain a configuration that prioritizes neural signaling efficiency over waste clearance. The interstitial spaces between neurons are relatively compressed, and the flow of cerebrospinal fluid through the paravascular channels of the glymphatic network is limited. During deep sleep, this configuration reverses. Glial cells contract. The interstitial space expands by as much as 60% in some estimates, and cerebrospinal fluid flows through the brain tissue with a completeness that allows the metabolic byproducts of a day of neural activity to be flushed from between neurons and directed towards systemic clearance pathways. Among the most critical of these byproducts are amyloid beta and tau proteins, the molecular accumulations whose progressive deposition in brain tissue, when glymphatic clearance is chronically insufficient, is directly associated with the neurodegenerative processes that represent the most feared cognitive consequences of. In the aging body, the architecture of sleep itself changes in ways that directly compromise both of these critical nocturnal processes, reducing the proportion of time spent in the deep slow-wave stages, where growth hormone secretion and glymphatic clearance are most active, fragmenting the continuity of the sleep cycle with the more frequent nocturnal awakenings that characterize aging sleep biology, and diminishing the overall restorative quality of the hours spent in bed to a degree that accumulates across months and years as a biological deficit whose consequences extend from the gradual loss of muscle mass to the progressive compromise of the neural. There is a molecule produced deep within the brain, in a structure so small it weighs less than 1/10 of a gram, whose influence over the sleeping body is so far-reaching, so architecturally fundamental to the quality and completeness of every night's rest, that its progressive decline across the decades of aging may be one of the most significant and least discussed contributors to the deteriorating sleep that so many older adults accept as simply the inevitable texture of life past 60. That molecule is melatonin, and the pineal gland that produces it, tucked within the geometric center of the brain between the two hemispheres, performs a regulatory function whose biological importance is entirely disproportionate to its modest physical dimensions. Understanding what melatonin actually does, why its production diminishes so significantly with age, and how certain extraordinary foods can support the pathway through which the aging brain still attempts to produce it, is to understand one of the most practically actionable nutritional insights available to any senior seeking to recover the sleep quality that advancing biology has quietly and progressively eroded. Melatonin is not simply a sleep hormone in the reductive sense that popular health culture has sometimes reduced it to, a chemical switch that turns sleep on when darkness arrives and off when light returns. It is a chronobiological signal of remarkable sophistication, synthesized by the pineal gland from serotonin in response to the absence of light detected by specialized retinal photoreceptors, and broadcast through the bloodstream to virtually every cell in the body, where it synchronizes the timing of local biological clocks with the external light-dark cycle that evolution has used to organize biological activity across hundreds of millions of years of life on a rotating planet. When melatonin rises in the evening, it does not merely signal sleepiness. It coordinates a cascade of hormonal, metabolic, and neurological shifts that prepare the body for the specific biological work of the night, including the reduction of core body temperature that facilitates sleep onset, the suppression of cortisol that allows the stress axis to rest, the modulation of immune activity toward the repair and consolidation functions that nocturnal biology prioritizes, and the orchestration of the sleep stage architecture through which growth hormone secretion, glymphatic clearance, and memory consolidation are temporally organized into the precise biological sequence that genuine restoration requires. In a young, hormonally robust pineal gland, this melatonin synthesis occurs with a reliability and a magnitude that produces the deep, sustained, multi-stage sleep whose restorative completeness most older adults now remember but no longer consistently experience. But the pineal gland undergoes a specific and progressive deterioration with age. Calcification of the glandular tissue reduces the number of functioning pinealocytes, the cells responsible for melatonin synthesis, and the enzymatic activity of the remaining cells declines in ways that reduce the peak nocturnal melatonin concentration achieved by an older adult to a fraction of what the same individual produced in their younger decades. This is not a minor hormonal fluctuation. It is a fundamental erosion of the primary chronobiological signal that organizes the sleeping body's restorative architecture, and its consequences for the sleep of people over 60 are specific, measurable, and experienced nightly by a majority of the senior population without any awareness of the underlying biological mechanism responsible for what they are. The kiwi fruit enters this biological story through a pathway whose scientific support has strengthened considerably in recent years, and whose practical implications for senior sleep nutrition deserve a prominence they have not yet consistently received. The kiwi is among the most concentrated whole food sources of serotonin, the neurotransmitter that serves as the immediate biochemical precursor from which the pineal gland synthesizes melatonin through a two-step enzymatic process, requiring first the acetylation and then the methylation of the serotonin molecule. When dietary serotonin from kiwi is absorbed through the gastrointestinal tract, a portion enters the systemic circulation and crosses into the central nervous system, where it contributes to the pool of serotonin available for pineal melatonin synthesis, supplementing the endogenous serotonin produced from dietary tryptophan through the metabolic pathway that the aging brain pursues with diminishing enzymatic efficiency. The vitamin C content of kiwi fruit supports this pathway at an earlier enzymatic step, the hydroxylation of tryptophan to produce the serotonin precursor. 5-hydroxytryptophan requires the cofactor vitamin C, and adequate vitamin C status therefore directly influences the efficiency with which the entire tryptophan to serotonin to melatonin biosynthetic chain operates in the aging brain. A fruit that simultaneously provides serotonin substrate, enzymatic cofactor support for serotonin synthesis, and antioxidant protection for the neural tissues performing these conversions is not offering a single nutritional contribution to sleep quality, but a layered, mechanistically coherent biological support for the entire pathway, whose Tart cherry extends this nutritional sleep architecture support through a mechanism that is distinct from and complementary to the kiwi's serotonin pathway contribution, the direct provision of dietary melatonin itself, present in measurable quantities in Tart cherries, in a form that is absorbed through the gastrointestinal tract and detectable in circulating blood in the hours following consumption. The melatonin content of Tart cherry, while modest in absolute terms relative to pharmacological melatonin doses, operates within a biological context that maximizes its effectiveness. Accompanied by anthocyanin compounds that reduce the oxidative and inflammatory burden on the pineal gland and the neural tissues involved in sleep regulation, and consumed at a time of day when even modest additions to the declining endogenous melatonin production of an aging pineal gland can meaningfully influence the magnitude and timing of the nocturnal melatonin rise that initiates the sleep architecture sequence. Research examining sleep parameters in older adults consuming Tart cherry juice before bed consistently demonstrates reductions in the time required to fall asleep, increases in total sleep duration, and improvements in sleep efficiency that reflect the combined biological contribution of dietary melatonin, melatonin precursor support, and anti-inflammatory neural protection operating together on a sleep system whose own capacity for these functions has been quietly diminished by there is a window of biological opportunity that opens each night in the sleeping body of every person over 60, a window created by the nocturnal surge of growth hormone that represents the single largest anabolic hormonal event in the body's 24-hour cycle, and that window, for the overwhelming majority of older adults, closes each morning having been only partially utilized, not because the hormonal signal was absent or inadequate, But because the amino acid substrates that signal was attempting to direct toward muscle repair and protein synthesis were simply not available in sufficient quantity to allow the nocturnal anabolic program to proceed with anything approaching its full restorative potential. This is the nutritional gap that a pre-sleep serving of Greek yogurt addresses with a biological precision so specific, so mechanistically well-matched to the exact physiological challenge it is targeting, that understanding it completely changes the way any thoughtful person over 60 ought to think about the hours between their last meal and their first morning movement. The overnight fast is the longest period of amino acid withdrawal the body experiences across any continuous 24-hour window, typically spanning 6 to 8 hours, during which no new dietary protein enters the digestive system, no new amino acids are absorbed into the bloodstream, and the muscles must sustain whatever protein synthetic activity the hormonal conditions of sleep are attempting to drive from whatever circulating amino acids remain from the last meal consumed hours earlier. In a younger physiology, this overnight amino acid deficit is managed with relative biological efficiency, lower basal protein breakdown rates, higher anabolic sensitivity to available amino acids, and a more responsive growth hormone secretory system together ensure that the overnight fasting period, while not nutritionally optimal for muscle maintenance, does not produce catastrophic net protein loss across the sleeping hours. In a body over 60, each of these compensatory capacities has been measurably compromised. Anabolic resistance has elevated the amino acid threshold required to activate muscle protein synthesis. Basal protein breakdown rates have risen relative to synthetic rates, and the growth hormone secretory response is both smaller in magnitude and less reliably translated into downstream muscle anabolic signaling through the insulin-like growth factor 1 pathway, whose sensitivity also declines with advancing age. The result is an overnight protein balance equation that is fundamentally less favorable in the aging body than in the younger one, and that tilts progressively further toward net protein loss across the sleeping hours, as these overlapping anabolic deficits compound one another in a physiology whose compensatory reserves have been quietly diminishing across the decades. Every morning that a person over 60 wakes without having provided their muscles with adequate overnight amino acid support is a morning that begins with a small but real increment of net muscle protein loss that daytime nutrition and exercise will struggle, and frequently fail, to fully reverse. Night after night, across the months and years of a senior's life, this accumulated overnight deficit contributes to the progressive sarcopenic decline that no amount of well-intentioned daytime dietary effort can completely compensate for when the longest fasting window of the day is left nutritionally unaddressed. Casein protein, the slow-digesting dairy protein that comprises the majority of the protein fraction in Greek yogurt and cottage cheese, addresses this overnight biological gap through a digestive kinetic profile that is uniquely suited to the temporal demands of the nocturnal anabolic window in a way that no other commonly available protein source replicates with equivalent precision. When casein enters the acidic environment of the stomach, it undergoes a process of coagulation, forming a semi-solid gel-like structure that dramatically slows the rate at which proteolytic enzymes can access and cleave its peptide bonds, producing a gradual, sustained, prolonged release of amino acids into the small intestinal lumen and subsequently into the portal circulation that maintains elevated circulating amino acid concentrations across a period of 4 to 6 hours following consumption. This is not merely a slower version of the rapid absorption that characterizes whey protein. It is a fundamentally different digestive pattern that produces a fundamentally different amino acid availability profile across the hours of overnight sleep, maintaining the leucine and essential amino acid concentrations required to sustain mTOR pathway activation and support muscle protein synthesis across the majority of the nocturnal window, rather than producing the sharp, brief amino acid peak of rapidly digested proteins that rises and falls within 2 hours of consumption leaving the remaining hours of the night in the same amino acid deficit that preceded. The practical consequence of pre-sleep casein provision through Greek yogurt for the aging muscle is not a theoretical improvement projected from mechanistic reasoning alone, but a finding that controlled research in older adult populations has examined with increasing rigor and consistent support. Studies comparing overnight muscle protein synthesis rates in seniors who consumed a casein-containing pre-sleep protein source against those who consumed no pre-sleep protein consistently demonstrate significantly higher rates of overnight muscle protein synthesis in the supplemented group, rates that translate across weeks and months of consistent pre-sleep protein provision into measurably better preservation of muscle mass, muscle strength, and physical performance outcomes in a population for whom these variables determine not merely athletic capacity, but the functional independence, fall resistance, and metabolic health that define the quality of daily life past 60. Greek yogurt delivers this casein protein in a whole food matrix that simultaneously provides calcium for the bone muscle unit, phosphorus for cellular energy metabolism, and the probiotic bacterial cultures whose influence on gut microbiome health and systemic inflammatory tone adds a further dimension of biological benefit to what is, at its most essential, a remarkably simple and remarkably powerful act of nutritional intelligence. Eating a small bowl of yogurt before bed and giving the sleeping body's most ambitious biological program the raw materials it has been asking. There is a mineral so quietly fundamental to the biological transition between waking and sleeping that its deficiency, extraordinarily common in adults over 60 and almost universally unrecognized by the people living inside it, may be one of the most significant and most correctable contributors to the deteriorating sleep quality that the aging population accepts as inevitable when it is, in meaningful and measurable part, nutritional. Magnesium occupies a position in the nervous system's regulatory architecture that no other mineral replicates, not merely as a cofactor in hundreds of enzymatic reactions, not merely as a structural component of bone and cellular energy metabolism, but as a direct molecular participant in the neural processes of inhibition, calming, and deactivation, through which the waking brain releases its grip on alertness and permits the body to enter the deeper sleep stages where its most important restorative work is performed. To understand what magnesium does in the sleeping nervous system and what its deficiency quietly prevents is to understand why a small handful of almonds or pumpkin seeds consumed before bed is not a trivial nutritional gesture, but a biologically targeted intervention for one of the most pervasive and most underaddressed obstacles to genuine sleep restoration in older adults. The GABA receptor system is the primary inhibitory neurotransmitter pathway in the central nervous system, the molecular mechanism through which the brain achieves the neural quietening that sleep requires, suppressing the excitatory signaling of the waking day and creating the neurological conditions in which consciousness can relinquish its hold on active processing and permit the orchestrated unconscious biology of sleep to proceed. Magnesium is a direct modulator of GABA receptor sensitivity. It binds to specific sites on GABA A receptors and enhances the receptor's responsiveness to GABA itself, amplifying the inhibitory signal and facilitating the neural calming that the transition from wakefulness to sleep demands. In a nervous system adequately supplied with magnesium, this GABA-mediated inhibition operates with the efficiency that allows sleep onset to occur without the prolonged wakefulness, the racing thoughts, the inability to release the day's cognitive content that so many older adults experience as simply the way their minds behave at night, when in biological reality, they may be experiencing the neurological consequences of a mineral deficiency that is preventing their inhibitory neurotransmitter system from performing the very function for which it. Beyond the GABA pathway, magnesium regulates the N-methyl D-aspartate receptor, the primary excitatory glutamate receptor in the central nervous system, through a mechanism of voltage-dependent channel blockade that is one of the most elegant examples of mineral-mediated neural regulation in the entire nervous system. At resting membrane potentials, magnesium ions physically occupy the channel pore of the NMDA receptor, preventing the calcium influx that would otherwise maintain neural excitation and prolonging the state of relative neural quiescence that facilitates the transition into deeper sleep stages. When magnesium is deficient, this channel blockade is less complete. NMDA receptor-mediated excitation is less effectively suppressed, neural arousal thresholds are lower, and the sleeping brain is more susceptible to the excitatory stimuli that produce the nocturnal awakenings, the light-fragmented sleep, and the early morning waking that characterize the sleep of magnesium-deficient older adults, with a consistency that the research literature has documented across multiple study populations, and that clinical experience in the aging population confirms with almost monotonous. The hypothalamic pituitary adrenal axis, the stress hormone system whose nocturnal suppression is a prerequisite for the cortisol reduction that allows the sleeping cardiovascular system to achieve its necessary blood pressure dip, and the sleeping brain to enter the growth hormone secretory stages of deep slow-wave sleep without hormonal interference, is directly regulated by magnesium at multiple points in its signaling cascade. Magnesium inhibits the release of adrenocorticotropic hormone from the pituitary gland, reducing the downstream cortisol secretion that would otherwise maintain the physiological arousal state incompatible with deep sleep. In a body over 60, where the stress axis is already prone to the chronic low-level activation that characterizes aging biology, where baseline cortisol is elevated relative to younger decades, and where the nocturnal cortisol suppression required for deep sleep occurs less completely and less reliably, the magnesium-mediated inhibition of HPA axis activity is not a supplementary benefit, but a primary mechanism through which adequate magnesium status directly enables the hormonal conditions that restorative sleep. The cardiovascular easing that must occur during sleep, the nocturnal dipping of blood pressure, the reduction of heart rate, the lowering of arterial tone that allows the sleeping heart to recover from the demands of the waking day, depends on magnesium's role as a physiological calcium channel antagonist in vascular smooth muscle. Magnesium competes with calcium at the smooth muscle cell membrane, reducing the degree of vascular contraction and promoting the arterial relaxation that lowers peripheral resistance and allows blood pressure to achieve its necessary nocturnal reduction. In a senior population where hypertension is prevalent and where the nocturnal blood pressure dip is frequently blunted by the arterial stiffening of advancing age, the magnesium provision of a small pre-sleep serving of almonds or pumpkin seeds, two of the most magnesium-dense whole foods available, contributes to the vascular easing that the sleeping cardiovascular system requires to perform its own nocturnal restoration within the reduced pressure environment that cardiac recovery after 60 genuinely. There is something almost poetic about the fact that one of the most biologically useful foods a person over 60 can eat before bed is also one of the most ordinary, most universally available, most completely unassuming foods in the entire human dietary landscape. The banana sits on kitchen counters and fruit bowls across the world without any particular mystique, without any premium price, without any of the nutritional celebrity that has been attached to far more exotic foods whose biological relevance to the aging body is, in many cases, considerably less specific and considerably less well supported than the humble, yellow, entirely unremarkable fruit that most seniors walk past without a second thought on their way to close the kitchen for the night. And yet the banana, understood in the context of what the aging body actually needs across the hours of overnight sleep, reveals itself to be one of the most multidimensionally targeted pre-sleep nutritional interventions available to any person over 60, addressing simultaneously the electrolyte imbalances that fragment sleep through nocturnal cramping, the tryptophan pathway that supports the melatonin synthesis, the aging pineal gland can no longer sustain independently, and the blood pressure modulation that allows the sleeping cardiovascular system to achieve the nocturnal dip that cardiac recovery after 60 specifically requires. Nocturnal leg cramps, the sudden, involuntary, often intensely painful muscular contractions that awaken older adults from the deeper sleep stages with a frequency that research consistently finds to be significantly elevated in the over 60 population, represent one of the most disruptive and least discussed contributors to the fragmented sleep architecture that characterizes aging biology. Their mechanism is rooted in the electrolyte economy of the muscle cell, specifically in the balance of potassium, magnesium, and calcium across the muscle cell membrane that determines the controlled repolarization following contraction, the process through which a contracted muscle releases its tension and returns to its resting length. When potassium concentration inside the muscle cell falls below the threshold required for efficient membrane repolarization, as it does with increasing frequency in older adults whose renal potassium handling has become less precise, whose dietary potassium intake has often declined with the reduced appetite of advancing age, and whose medication regimens frequently include diuretics that accelerate potassium excretion. The repolarization process is disrupted. The muscle membrane remains in a state of sustained depolarization, and the uncontrolled, prolonged contraction of a cramp is the physiological consequence of an electrolyte deficit that adequate potassium provision would have prevented. A banana consumed before bed, providing a meaningful contribution to the muscle cell's potassium economy across the overnight fasting hours when no further dietary electrolyte provision will arrive, does not merely address a painful inconvenience. It protects the continuity of the deep sleep stages during which growth hormone secretion, glymphatic brain clearance, and muscle protein synthesis are performing their most critical nocturnal work from the electrolyte-driven interruption that so many older adults have accepted as the unpredictable and unpreventable texture of nights past 60. The tryptophan content of the banana adds a further dimension of pre-sleep biological relevance that operates through the same melatonin biosynthetic pathway addressed by the kiwi fruit and tart cherry discussed earlier in this conversation, but with an additional mechanism that makes the banana's tryptophan contribution particularly bioavailable for conversion to serotonin and subsequently to melatonin in the aging brain. Tryptophan is an amino acid that competes with other large neutral amino acids for transport across the blood-brain barrier through a shared carrier mechanism, meaning that when tryptophan is consumed alongside significant quantities of competing amino acids from protein-rich foods, the proportion that successfully crosses into the central nervous system where its conversion to serotonin can occur is reduced by competitive inhibition at the carrier. The banana's modest carbohydrate content, delivering a gentle, gradual glycemic response rather than a sharp glucose spike, stimulates a modest insulin release that preferentially drives the competing large neutral amino acids into peripheral muscle tissue for protein synthesis, reducing their plasma concentration and thereby increasing the relative proportion of tryptophan available for blood-brain barrier transport and central serotonin synthesis. The banana effectively clears the competitive landscape for its own tryptophan, making its melatonin pathway contribution more biologically effective than the same tryptophan quantity consumed in a different nutritional context would produce, a detail whose practical significance for sleep quality in older adults is both real and entirely invisible to anyone approaching the banana as merely a potassium source, rather than as the multi-mechanistic pre-sleep nutritional instrument that the biology reveals it. The potassium content of the banana also participates directly in the blood pressure story of the sleeping cardiovascular system, contributing to the natriuretic effect through which adequate potassium intake encourages the kidneys to excrete sodium rather than retain it, reducing the osmotic blood volume expansion that drives nocturnal blood pressure above the dipping threshold that cardiovascular recovery during sleep requires. In a senior population where hypertension is prevalent, where the nocturnal blood pressure dip is frequently blunted by the arterial stiffening and sodium retention of aging biology, and where the sleeping heart's capacity for genuine recovery from the day's hemodynamic demands depends on achieving adequate pressure reduction during the overnight hours. The potassium contribution of a pre-sleep banana is not a peripheral cardiovascular gesture, but a meaningful nutritional participation in the vascular conditions that determine how well and how completely the sleeping heart restores itself across the hours that separate one day's demands from the what the full story of these four pre-sleep foods ultimately reveals is something that extends beyond any individual nutrient or any single biological mechanism. It reveals that the sleeping body of a person over 60 is not a passive biological landscape that simply receives whatever the day has provided and makes do with what remains. It is an active, demanding, nutritionally specific biological system, performing work of extraordinary importance across the hours of darkness, and it deserves to be nourished with the same scientific intelligence and the same deliberate intention that the most health-conscious among us bring to the meals of the waking day. The kiwi supporting the melatonin architecture that age has eroded. The tart cherry adding direct dietary melatonin where the pineal gland can no longer provide it independently. The Greek yogurt sustaining the overnight muscle protein synthesis that growth hormones' nocturnal secretion requires substrates to drive. The almonds and pumpkin seeds providing the magnesium that the nervous system needs to release the day and descend into the restorative depths, and the banana maintaining the electrolyte integrity and tryptophan availability that protect both the continuity and the quality of the night's most biologically valuable hours from the deficits that advancing age has quietly introduced into the overnight physiology of a body that deserves every opportunity to rest, repair, and rise tomorrow with the biological resources that a well-nourished sleep has always been capable of. If this conversation has given you something genuinely valuable, a new understanding of what your body is actually doing while you sleep, a scientific clarity about why these four ordinary foods carry such extraordinary nocturnal biological significance, and a renewed sense that the quality of your rest is not fixed by age, but responsive to the nutritional intelligence you bring to the hours before bed, then carry that understanding forward and join the community that keeps this science alive and growing every single week. Come and find Dr. Martin Hale on YouTube, your dedicated home for senior health, where every video is built on exactly the same foundation as this entire conversation: rigorous science, compassionate delivery, and an absolute unwavering belief in the capacity of the human body to repair, restore, and renew itself at every age when it is given what it genuinely needs. If this moved you, if it gave you something worth keeping, press the like button, because that single action tells the algorithm to carry this conversation to another senior who is lying awake tonight without understanding why, whose muscles are losing ground overnight without knowing it can be addressed, whose sleep is fragmenting without realizing that the biology behind it and the nutritional response to it are both within reach, and your like may be the reason they finally find Dr. Martin Hale and the science that could change the quality of every night that follows. Share this with someone you love who deserves better sleep, stronger mornings, and the understanding that the hours of darkness are not empty, but full of biological possibility, that the right four foods can help their aging body finally, fully, and gratefully